So the vocabulary words we covered so far are words like biogeography, which relate to what life is found where. What is the geography like? In other words, is it hilly? Is it rocky? Is it soil? Is it salt water? Is it fresh water? Is it dry? Is it cold? But also, what life is found there? Is there fungi found there? Bacteria, mammals, reptiles? What types of plants? So it's what is the geology like, but it's also what is the life that exists there. Chronometric dating, we've looked at how we can look at chrono means time, and so we can date different objects, different fossils, different rocks with different methods. We can put them within a time range, within a certain range of age, say a minimum of this and a maximum of that, with some degree of error, but relatively speaking, we can get some pretty good data, both absolute and relativistic data, right? Common ancestor, that's that idea that we have as we go back in lineage, as we look at who begat who, going back beyond our most recent history, back millions of years or thousands, hundreds of thousands of years, millions of years, we can look at what organisms accrued enough mutations and were isolated enough to become a different species, and what was the origin of that species. Which came first, the chicken or the egg, right? Today we know it's the egg. Some bird species collected enough mutations that one of their eggs or a few of their eggs started to develop the characteristics of a chicken. So those are common ancestors. Comparative anatomy, we've done that a lot. We've looked at horses, we've looked at the ancestry of horses, we've compared the hands of bats and birds and humans, and we use this anatomy, these anatomical similarities and differences to categorize organisms into different groups. We've looked at things like homologies. We've said, okay, this hand has similar bones to this hand, and even though one's used as a flipper, the other one's used as a wing, and the third is used to climb, they're all pentadactyl. They all have 5 fingers, so they're related. All these vertebrates are related in some way. An epoch is a period of time, geological time. Evo Devo, right? Evolution and development. When we look at things like the Hox genes, fossils are remnants of ancient living organisms. Generally, they're either impressions or they're mineralized remains. So bones that have long ago broken down, but the minerals, as the water gets absorbed with all the minerals, those minerals start to form rock within their same structure. So the structure, the shape of the thing stays the same, but it's no longer bone, it's some type of rock. So that's a mineralized fossil, and there's also impressions, footprints, and body prints that are left over after something gets buried or crushed. The fossil record, right? Where we find fossils, how deep are they in the strata? What age are they? This is a record of how life has changed on the planet over time. Geologic era is another measurement of time, very long period of time, and you can look at geologic period, geologic era, and epoch. Look up all 3 and know which one's bigger, which one's smaller. Geologic time scale. Obviously, this is a really big idea, even though it seems kind of simple. Okay, it's a time that we're talking about epochs and periods and eras, and they're talking thousands and millions and millions and millions of years. But when we're talking about geologic time scale, the hard part is really grasping the length of time that you're dealing with. When you're dealing with a geologic time period, when you and I talk about time, we talk about seconds and minutes and days and weeks, and we've been stuck in our homes now for about a month. We might be stuck here for another couple months. We think this is a long time, but as far as the geologic time scale is concerned, it's not even a blink of an eye. The Earth's time, the time it takes to build a mountain, for a valley to form, for a fault to move great distances, for continents to drift, are in the millions of years. And so the geologic time scale has to be broken down into units that are much bigger than what we're used to, and it are difficult to comprehend, to be honest. Homologous structures are structures that are similar between different species, but that have evolutionary linking, right? So we've inherited from a common ancestor, but we have these homologies, or two things have homologies that they inherited from a common ancestor. And we've looked at the horse, and we've looked at the various characteristics that you can look at, and if you go back and look at the charts that scientists have put together on comparing different structures, different phenotypes, and looking at homologies that they share. The homologous structures that we really looked at were, the hand, the five-fingered hand, and how different species have either lengthened or shortened the bones in order to use them in different ways. Hox genes are those genes that are involved in the development of the organism, whose order in the genome impact their expression. And so one gene later on, just because it's later in the genome, further downstream of another gene, it can actually turn the first gene off. And depending on where it's located and what tissue certain genes are turned on and off, it makes things bigger or smaller. It could make they could change all kinds of structures depending on where these genes are found and how many copies of these genes there are. So Hox genes are very interesting, and the details are complicated, but they're involved in the development of vertebrates. I not even say vertebrates. They're involved in the development of creatures, because remember, they were first described in fruit flies, which are invertebrates, right? They don't have a backbone. They have an exoskeleton. They don't have bones at all. They have an exoskeleton. So Hox genes have been found in some of our earliest ancestors and are found in us. In multicellular life, you have to have something that guides the organization, the differentiation, the specialization, right? Those are words that all mean the same thing, of cell types, right? So you have to have something that says, okay, I need to make an eye, I need to make a brain, I need to make skin. And the Hox genes help in that decision making. They can be turned off by environmental factors. There are things like small RNAs. There are these long non-coding RNAs. They can interact with each other and turn each other off. There's all kinds of regulation that have been described in the Hox genes that are important to the development of the creature, to see a creature go from a single cell to a trillion cell multicellular organisms with different tissues and different organs and organ systems, all because certain genes can be turned off and others on. Regulation. Hox genes are really the example of gene regulation that really help you understand how development of an organism happens, but then we can look at what characteristics of these Hox genes we share with other organisms, and which Hox genes and their character and their controls are shared among different organisms, to look at evolutionary relationships. Molecular clocks, we looked at mutations happen at a specific rate And if mutations happen at a specific rate, anything that happens in a specific amount of time, over and over and over again, constantly, we can use them as clocks. We can use atomic atoms breaking down at a specific time. We can look at mutations happening in a very specific amount of time. But molecular clocks are notoriously difficult to get really straightforward answers on. There's all kinds of things that can make it a little more difficult. You could have an environmental impact causing faster mutation, or other environmental impacts that might slow things down, even in non-selected or neutral genes that have that are only impacted by genetic drift. Molecular clocks are difficult to really pin down and have consistent data from. However, it is a source of data that we can look at and try to use as part of the evidence to put together a big picture, even if it is a little fuzzy. Paleontology is a study of the fossil record when it comes to prehistoric life before humans, right? So anthropology is a study of the human history. Paleontology is looking at the evolutionary history of ancient life. And so you're going to do a lot with fossils. They're dinosaur hunters would be a paleontologist. Phylogeny is the study of how organisms evolve. What are the various links between the redwood tree and the pine tree, or the moss and the lichen, or this bacterial strain and that bacterial strain. We've done a lot of that with this coronavirus, this new coronavirus that's come out. We know that this COVID-19 is in fact a coronavirus. So it's in a family of viruses. We know that it had mutated from some previous virus, like flu-like virus. We know that because of its characteristics, right? So we do this kind of comparison even when it comes to a virus. We know that this COVID-19 is a coronavirus because of the similarities it has with the flu and with HIV, where you have little spikes that come off, little like hammer-looking spikes that stick out of this little sphere. And because of that, we're going to say that they belong to a family and must have inherited it from a common ancestor. So this idea of phylogeny, or the study of evolutionary history of an organism, can really help us. It can help us, in this case, fight a disease. Radiometric dating is part of that chronological dating. Radiometric dating gives us a relative time period. It gives us it's an absolute time period. It's not relativistic like it is with the strata, right? We can't just, you look at a rock strata, you might say this one's older than that one because it's underneath the one that's on top. That's relativistic. Radiometric is called absolute because we have a time period that we can say it's about 1 million years old. Now, the reason I always say it's relatively accurate is because you don't really have a specific period of time. You can't say this rock was made in 2000 BC. You can say it was about 50,000 years old. This thing is about 2 million years old. You can't really come up with a specific time when it comes to radiometric dating, but it is an absolute time period. We know that it can't be any younger than this or any older than that because of how much radiation, how much of the radioactive decay product is left in the material. Relative dating is exactly what I just described, right? Where you have the rock strata, one's older than the other, but you don't have any real time period. Rock strata, again, is those layers of rock that we've discussed, that we've gone over and drawn and looked at, and we've talked about all the different things that move these rock strata around, that subduct them, that push them up, that slide them next to each other, that could flip them, and when a volcanic eruption occurs, you can find these rock stratas, the strata disrupted, but if they're not disrupted, they were layered down one on top of the other on top of the other over a long period of time. So the one on top is the youngest, the one on the bottom is the oldest. We call that kind of relative dating using rock strata. Transitional fossils are fossils that are in between two species, right? And the great example that we looked at with them is the horses. How many transitional fossils did you look at in that chart about when it came to horses? You can see those four toes in the fossil record. You saw them lose toes throughout their history, and you saw their snouts become longer and the single toe become longer and the horse become taller and bigger and more stable. You saw the transitions in the fossil record. Then vestigial structures are those structures that don't have a purpose or don't have a very big purpose anymore. And we've looked at a number of those vestigial structures. We said, things like tonsils and appendix, and things like the hip bone in a whale, the leg bone in a whale. Why does a whale have a leg bone? It's not part of its flipper. It's not part of. I shouldn't say leg bone. I should say rear limbs. It doesn't have rear limbs. It doesn't have rear flippers. They don't use them anymore, but they still have the bones. They have no real purpose for being there. And there's several of these examples that we had discussed when it came to vestigial structures. So each of these vocabulary terms I think we've described pretty well, some better than others, I must admit, but hopefully you've been able to get something out of this workbook, this section of the workbook. Now we're going to go ahead and move on to the next section.